quite a lot of the voxels have kind of quite a lot of yellow stuff in them, and you can see that quite a lot of the voxels are quite this kind of mid-scale gray here. Okay, so what's the end result? The end result is that we have different activation patterns for different orientations. And this is basically the thing that makes pattern-based fMRI possible, the thing that makes multivoxel pattern analysis possible. So different stimuli induce different patterns across multiple voxels. So if you were to just look at one voxel at a time, many of these voxels are individually not very informative at all. Okay, so say I take this one here that's like 2 down, 2 across. Okay, it's kind of quite active-ish for many different orientations. So if I see this voxel is kind of active-ish, that doesn't really tell me anything about what the orientation is, because all of these orientations actually make this particular guy here kind of have a certain degree of activation. And this is a very, very common thing in any brain region in which you have multiple different stimuli are all activating the same region. They're all plotted on top of each other. They're all activating on top of each other. So all of them are going to activate this region. So if you do a standard analysis of just saying, to what degree does this region light up, it's not really going to give you a very different answer for any of the different stimuli. So for instance, say you're interested in how people perceive the difference between one speech sound and another speech sound. If you just say, show me the activation for when someone's hearing bar versus da, bar's going to activate the auditory cortex, da's going to activate the auditory cortex. If you just look at activation, you won't see any difference. Same with orientation. This is the reason why everyone was kind of very pleasantly surprised and impressed when this paper came out, because everyone thought, there's no way you're going to be able to tell what orientation someone's looking at, because no matter what orientation you're looking at, it's going to light up their visual cortex. So if you say, they looked at this, yep, their visual cortex lit up. Yep, they looked at this, yeah, their visual cortex lit up. But more to the point, it's going to light up to more or less the same degree. It's not like our brains, well, it may actually be that our brains are very, very slightly more sensitive to what's called cardinal orientations, which are like these ones. But in general, any orientation is going to light up your visual cortex. It's not like we're blind to, these vertical lines look super intense to us, but diagonal lines look kind of dim and fuzzy. We can see them just as well as we can see the vertical lines, even if there's a very, very slight extra sensitivity difference to the canonical ones. So all of these, so if you just look at the activation of a particular voxel, they're all making it active, but it's the pattern that differs. This is really, if there's a single thing that you need to kind of understand in order to understand why kind of looking at multivoxel pattern activations is a worthwhile thing to do, or why it's any different from just a standard fMRI analysis, this is the reason. It's because if you were to count up how much gray stuff is there in this 4x4 grid for all of these different scenarios, the answer is it will be quite a lot, quite an equal amount for many of them, right? Like this one, this one, this one, this one. They all look like the total amount of gray stuff there is about the same, but the pattern's different. So I'm probably kind of flogging a dead horse on that one, but that was really when Haxby in his 2001 science paper, Haxby and colleagues first did this kind of analysis, that was their crucial insight. And it's not the kind of technically complicated insight. It doesn't require math. It doesn't require some fancy equations to see that these patterns are different. It just requires asking this very basic question: hey, why are we just looking at all the voxels one at a time? Why not look at the pattern of them? And that seems like such a basic question that you might think, well, that's just like blindingly obvious. It wasn't blindingly obvious at all, and it was a huge, huge breakthrough when this happened, because it enables totally different types of questions to get asked. So, Adam, yes?
So these patterns emerge over the mean of many trials.
Right.
So on a trial-by-trial basis, you could have any pattern
right.
Available. So this wouldn't help us on a trial-by-trial basis, just as a mean across all the runs, I guess, at the end of the experiment?
That's a very, very good question. Well, it depends how noisy each individual trial is. And actually, one of the things that we're going to do in a minute is kind of add some fake noise to this. So, and this is actually an open question. Okay, so I mean, each individual trial, if you've got tons and tons of noise and, you know, not that much signal in each individual trial, then you ain't going to be able to get much from each individual trial. But it depends, right? I mean, so there are, in fact, some studies saying that, you know, if you kind of do things right, you can get some information from an individual trial. But in general, you're going to get a much better signal if you have multiple trials all averaged together. And that's true, you know, that's not just true for imaging, that's basically true for every kind of measurement ever, really. You know, it's the same reason why, you know, if you're doing an opinion poll, you poll 1,000 people instead of just asking 2 or 3 people. It's the same reason why, you know, if you're measuring your blood pressure, you don't just measure it once, you measure it multiple times, and then kind of take the average of those. So, yeah, so individual, but you know, the fact that you have to get lots of measurements in order to try and deal with noise doesn't mean that each individual measurement isn't giving you anything. It just means it's giving you just like a little bit, and you might be lucky and you might get one of the ones without that much noise in it, and you might not get that. I mean, if you go to the doctors, they actually, you know, they take your blood pressure once, right? And in fact, it's a well-known result that, in fact, you usually get a systematically biased estimate of people's blood pressure from that, because everyone gets a bit nervous when someone in a white coat comes and takes their blood pressure. So if you just have an individual reading, the chances are that your reading is going to have all kinds of problems in it, just like, you know, the single measurement that you get when you go to the doctor. However, if you have a single measurement and you go to the doctor and you know, your blood pressure is like really, really, really high on that measure, even after taking into account that everyone's measurement is a little bit high, then they're probably going to say, oh, we need to measure your blood pressure a few more times, you know, here's a home monitor, please go and take this, and maybe you need to take some medication. So there's some information you can get from a single reading, but how much, you know, it depends upon how good your measurement is and how fundamentally variable the underlying thing is that you're measuring. Now, fMRI is fundamentally quite noisy, so you don't get a great deal of joy from individual trials, but it doesn't mean that you don't get anything. Yeah, I think I saw another question. Okay, so we're actually going to kind of inject some fake noise into this in a little bit, because that's the problem with simulations. It's very easy to have a simulation that's incredibly clean and beautiful and doesn't actually have anything to do with the real thing that you're trying to simulate. But the key point here is that, you know, each of these different orientations makes a different pattern. And now the question is, how are we going to go from that pattern to trying to figure out what the orientation was? That's the decoding part. Okay. So, and we're going to use a very, very simple measure in this case. This is actually not what Kamitani and Tong did. They used these things called support vector machines, which we're going to talk a bit more about later in the class. But we were just going to look at correlation, which is actually what the Haxby paper did, and which actually turns out to work quite well in many cases. And correlation is literally just, you know, what's the correlation of this pattern with this pattern? You know, what's the correlation of that pattern with that pattern? So just, you know, there's a spatial correlation of these things. Does one tend to go up and down when the other one tends to go up and down? Okay, so so this is kind of, you know, part just like showing you how to do stuff in Matlab, and part showing you kind of how you would actually think about how different voxel activation patterns might arise. Okay, so now let's actually try doing some basic classification. So let's see what they've got here. Suppose we're given a pattern of voxel fMRI activation. Can we figure out which orientation gave rise to it? That's the question that the Kamitani and Tong paper addressed. We can do this by calculating the correlation of that pattern with each of our original measured activation patterns and see which one it matches best with. So this idea of actually correlating things with each other and just seeing which one gives the best match is a very, very, very simple way of doing classification, but actually is quite good. It actually works quite well in many circumstances. So these are our, you can think of this as our training data, okay? So this is, we know that somebody saw orientation 1 and we got this activation pattern. We know that when they saw orientation 2, we got this activation pattern. And now I give you some new activation pattern, and I say, which orientation do you think they saw? And then you can literally just say, okay, well, I'm going to take my target activation pattern, my test activation pattern in this case, and see how well does it correlate with this one, how well does it correlate with this one, how well does it correlate with this one, and whichever one it correlates the highest with, that's the answer I'm going to pick. So, you know, classification, a classifier algorithm can seem like a complicated or difficult thing, but what I just described is actually a classifier algorithm, and it's actually quite a good classifier algorithm in many cases. It's literally just pick the best match. And in fact, where match is defined by correlation, and in fact, in terms of how this might not even be a terrible model for how we do things in everyday life. You have a kind of lineup of potential of criminals. Who is the person who mugged you on Friday night? Was it this person, this person, this person, this person? There's actually psychologically all kinds of problems with suspect lineups like that, but forget about that for a second. But which of these people was the best match? Pick that person. Okay, and then you say, oh, I think it was person number 3. Okay? And that's kind of how we actually do things. It's not a terrible system, except for when you're doing lineups. That's the whole. There's a very famous psychologist of memory called Elizabeth Loftus who's done a lot of work on. There's other people who've done work on this too, but it turns out that eyewitness memory is much less reliable than people actually think it is. People have been let out of prison because of this kind of stuff. It's kind of a big deal. Yeah.
So you and I talked about this very briefly before, but when looking at single unit data, it's common for people to use nearest neighbor approach to decoding.
Right.
Is that ever used in fMRI, or is it more?
Well, this basically is nearest neighbor. Okay, well, it depends how you define nearest neighbor, right? Because nearest
Euclidean distance.
Yeah, the nearest neighbor has 2 aspects of it. It has nearest and neighbor, right? So near means near defined by some distance measure. Okay, and there's different ways of defining distance. So and this is what we would. This is what Tommy and I were chatting about a little while ago. But so in this case, the measure of distance is spatial correlation. But there's other ways of defining distance. So for instance, Tommy just mentioned this thing, Euclidean distance, which is basically, instead of, say I got 2 patterns, instead of looking at the correlation of this pattern with that pattern, I say, take the absolute gray level values and get the difference, the voxel by voxel difference in gray level values, and square that and add them together and get the square root of that. So that's called the Euclidean distance, which is basically if these were points in space, how far apart would they be from each other? And that's another pretty good measure of distance. And different distance measures are sensitive to different things. A kind of metaphor that I like to use for this is, imagine you've got a black and white photo, and you want to say, you've got 2 black and white photos, and you want to figure out, are these basically the same photo? Okay? So suppose I take one of these black and white photos, and I just add a certain amount of brightness to every single pixel in it. Right? I say everything's between 0, and I just add 0.5 to everything, and if it goes off the ceiling at 1, I just cap it at 1 or something. Okay? So I've still got the same photo now. It's just like a brighter version of that photo, right? It's like the brightness slider in iPhoto or something. Okay? But if you say, what's the Euclidean distance between this photo and the one that I've just added lots of brightness to, it says, oh, they're quite different because this, there's a difference of 0.5 between every single pixel. Okay? Now, it's actually still the same photo. Okay? Now, what does it mean to say it's the same photo? It means it has the same pattern. Okay? So one of the it really depends. There's actually times where that might be a useful thing to do, but in terms of, for the question of, is this a photo of the same thing, you can see why kind of looking at the absolute value of intensities might not be that helpful. So that's one of the reasons why correlation is, I'm just recapping what we already discussed about. This is one of the reasons why correlation is kind of helpful, because correlation gets rid of, the first thing correlation does is it gets rid of the mean of each image and just kind of looks at the pattern between them. So,
So I guess in single unit data, one thing that might be important is the gain modulation, right? And so I guess I'm wondering, I'm not used to thinking about fMRI, so I'm wondering with fMRI, is that ever a useful thing?
No,
Like,
definitely. Definitely, you're absolutely right. So, I've been talking about how, I've been putting a lot of emphasis on saying, just looking at average intensity of activation is not necessarily that helpful because, different, if you have lots of different stimuli which all activate the same area to more or less the same degree, you can't tell the difference between that. But there's other times when, and there's many cases, and this was in fact true for, the first 10 plus years of fMRI, and still many studies done like this for perfectly good reason. There's many cases when just the amount of average local activation intensity is a very useful measure. So if you want, for instance, to see, especially if the thing that's being coded is something that itself varies in intensity. Okay, so if you say, for instance, I've got, say you show someone like a flashing checkerboard, just an image that's just kind of flashing on and off, and it might be a low contrast flashing on, where it goes to kind of gray versus black, or a really high contrast one where it goes to black and white. Right? If you change the contrast on that and you just look at what's the activation intensity in primary visual cortex, you get basically a straight line. That says, so you're literally coding for contrast in primary visual cortex purely by activation intensity, which makes a lot of sense because contrast is itself something that kind of comes at different intensities. And there's all kinds of other things that don't have to be as low level that are kind of like that too. So, for instance, if you give someone, say, a one-back working memory task, and they're kind of having to remember the thing that came before, and it's kind of hard to do, but it's not that crazy. You get a certain amount of activation in their prefrontal cortex. If you give them a two-back task, and now it's much harder because they have to remember two things back, and they have to make sure they don't get it mixed up with the thing that was one back, and it's really hard to do a two-back task. Okay. Then you get more activation. You're three back, and they're really sweating now. Then you get more activation. So again, the intensity is actually quite directly related to the degree to which you're kind of, making that area work. So these are very, very reasonable questions. But if you were to, but this is something again where it's really a kind of overall level of intensity that's kind of what's being coded in the first place. But if you wanted to say, not how hard are they working in this memory task, but what is it that they're trying to remember? Are they trying to remember, a telephone number or a word or something like that? If I'm trying hard to remember a telephone number, it's going to light up my prefrontal cortex. If I'm trying hard to remember a word, it's going to light up my prefrontal cortex. If I just say, how lit up is my prefrontal cortex, will that tell me whether I'm remembering a word or a telephone number? It probably won't, because they're both lighting it up. And in fact, actually, people have done studies using pattern, including Frank Tong actually has done this one, using kind of pattern-based analyses, trying to say, can you figure out what's in someone's working memory? And it turns out you can to some degree by looking at kind of pattern differences. So this is in some ways not a very satisfying answer, but the question of, what's the best kind of distance measure to use? When does it make sense to look at patterns? When does it make sense to look at activation intensities? The answer is, it depends what question you're asking. But there's not a single right answer, unfortunately. It'd be easier if it was. For a lot of single unit stuff, if all of the neurons are suddenly have their baseline firing goes up a lot, that's an important thing, right? That's something that the brain would probably care about a great deal. So if I say, so that's really not the same thing as, I take a photo and I make all of the pixels a bit brighter, but it's still the same photo, right? If you took some set of neuron activations in the brain and you made them all, 30 spikes per second more active, that's not something which another part of the brain would just say, yeah, I don't really care about that, I just care about their distance from baseline. It'd be like, oh my word, this part of the brain's going crazy, this must be telling me something. So, it depends what you're interested in. But I would say looking at patterns is especially useful for when there's multiple different stimuli that are all activating the same region to more or less the same degree, which happens all the time. Okay, so how would you look at the correlations of these things with each other? Well, it turns out it's actually quite easy to do. And in Matlab, there's a command called car that looks at correlation. Okay, so we're going to look at the correlation of these things. Now, when you think about how you plot a correlation, typically a correlation is like a 2D scatter plot, right? Like this, and then there's some kind of cloud of dots. So that's, if it's two-dimensional, that's basically just taking one chain, one row of numbers and another row of numbers and just saying how do they correlate with each other. But what we have here are not just like sequences of numbers. What we have here are grids. And so the first thing you do if you want to calculate a correlation is you take your grid and you just turn it into a row of numbers. Now, of course, you actually lose something when you do that, right? You lose the spatial structure, the fact that it's a grid. So we can talk about that too if you're interested. The other ways of looking at this that kind of preserve the spatial structure there are, but they're kind of a bit more complicated. But a simple way of doing it is to say, let's just turn this thing into, let's just stretch this thing out into a single row of numbers, or a single column of numbers in this case, and take that one column, another column, and just correlate them with each other.
Why is it important to preserve the spatial structure? Like, why would that be better?
Well, because in this particular instance, it might not be that interesting, but the brain cares about spatial structure. So, we were just talking about how the brain has all these continuous maps in it, right? So, nearby parts of the brain tend, especially if they're part of a map, to code for nearby things. So if I take this, that's a very good question, by the way, thank you. If I take this 4x4 grid and I just stretch it out into a column, right? In the grid, this voxel here is kind of next to this voxel here, but in the column, they're not next to each other anymore. So if there actually is some real kind of continuity of function between this patch of brain and this patch of brain, I've lost that when I just stretch it out.
Why would you lose that? Couldn't you code that information into your array?
Yeah, you can try and there's actually there are ways of looking at relations between patterns that does try to keep some of the spatial structure in. Yeah. There's for instance, there's a thing called Earth mover's distance, which is kind of an interesting metric, but it's much more complicated to calculate that than just doing a simple correlation. If any of you happen to be interested in that, by the way, I actually wouldn't mind doing a study on whether Earth mover's distance is a good metric to use for neural data, but that's if any of you happen to be into that kind of thing. People in image processing use that a lot. But yeah, these are much more, as you can imagine, these are much more kind of complicated measures because, like you said, you have to encode in there somehow the spatial information. And then the question is, well, how do you encode that in there? And then you have different choices. Anyway.
So do you do a different pattern analysis when you keep it in a grid versus in a single one? Is that what you're saying?
Most, at the present moment, and I'm not saying for 1 minute that this is an optimal way to do it, which is actually why I'd be kind of interested in looking at this thing I just mentioned, if anyone is interested in that. Most pattern analysis at the moment basically just says, let's treat, step one, take your pattern, stretch it out into a vector. And it's really just for computational convenience that people do that. And because that actually kind of works okay, people haven't worried that much about the fact that you're losing some spatial information when you do that. But maybe people should worry about that, and actually that's kind of on my list of things I'd like to look at some point. Yeah.
I mean, doesn't something like a searchlight analysis actually kind of implicitly encode spatial information, right? Because you're only considering a pattern as a sort of spatially contiguous set of voxels.
That's right. So there's a sense in which it does and there's a sense in which it doesn't. We haven't talked about this yet, but there's a very popular method of doing fMRI analysis, which was invented by this guy Krieger-Skorter, whose name comes up a lot in the readings that I've assigned because he's done fantastic stuff, is that you basically go through the brain and kind of take a little local neighborhood, little local neighborhood, little local neighborhood, and work out the pattern differences or similarities within each of those neighborhoods. And then you kind of write a brain full of numbers, which is, what's the amount of pattern information in this neighborhood, this neighborhood, this neighborhood. So in that sense, each one of those individual steps is preserving some information about the local neighborhood. But then what's the first step within, typically, after having gotten that neighborhood, what's the first thing that gets done to it? You turn it into a vector. Yeah. So that's actually that's a very, very good point. That's a kind of midway, that's a midway solution. But it's still ignoring the spatial relationships within each individual searchlight. And then there's a different flavor of searchlight where you do searchlights on the surface of the brain instead of treating the brain as a 3D thing. There's all kinds of different approaches. And the answer to the question which one's the best is nobody knows yet. People are still trying to figure this stuff out. This is all kind of quite fresh and new. Okay, so for now we're just going to look at correlations. So let's look at some correlations. Okay, so so it turns out it's incredibly easy to calculate correlations in Matlab. So the first thing you have to do is you have to stretch things out into, remember, this does throw away some spatial information, but you just stretch things out into a single vector because that's just what correlation requires, this type of correlation. And in Matlab, the way you do that is just this kind of slightly mysterious looking command of just putting a colon after a matrix. So this grid is a 4x4 thing, but if I take that 4x4 thing on any matrix and I stick this little colon after it, it turns it into a column vector. So we stretch them all out, and then instead of having 8 different grids, we have 8 different vectors. And so this is our set of 8 different vectors, and then each one of these columns corresponds to the activation for orientation 1, activations for orientation 2, etc. And then and we're going to look at the correlation of each one of these columns with each one of the other columns. That's the correlation of each one of these maps with each one of the other maps. And that's what this line here does. So let's have a look at that. So here's the correlations of each pattern with the others. Okay. So remember, we've got 8 different orientations. Don't know what happened to this y-axis label here, that's weird. Okay, we've got 8 different orientations, and what does this say? This is a correlation matrix. You're going to see this kind of stuff in pattern-based fMRI analyses a lot too. So the entry 1, 1 means the correlation of the pattern listed by orientation 1 with the pattern listed by orientation 1, in other words, with itself, so that everything's perfectly correlated with itself. No surprise there, okay? So you have a diagonal of 1s. The diagonal is every entry of everything with it correlated with itself. Diagonal of 1s all down the middle. So that's one of the ways you can tell you're looking at a correlation matrix. It always has this diagonal of 1s all down the middle. And it will be symmetrical too, because the correlation of A with B is the same as the correlation of B with A. So notice there's this kind of bright strip down the middle. What this means is that the activation map listed by orientation 1 is perfectly correlated with itself, but it's quite well correlated with the nearby orientation. Remember, these orientations are numbered according to how far they are around the circle. So 1 is quite similar to 2, and it produces a quite similar orientation pattern. And it produces a little bit less similar orientation pattern for 3, and then by the time you've got an orthogonal orientation, they're very different. So this is basically saying that everything correlates perfectly with itself, quite well with its neighbors, and not very well at all with highly different orientations, which is exactly what you would expect. But real life isn't that clean. Okay, so here's where you add in some noise. I'm actually just going to, we only have a couple of minutes, so I'm going to skip that. Here's where you add some noise. You just literally add some noise to those matrices, and you end up getting slightly less good correlations, which is kind of what you'd expect. Okay, how do you turn this into a classification? It's really actually not that difficult. Okay, so we've got a whole bunch of correlations with each other. We call those noisy cores in this case. Okay? And you say, for any given orientation, which one is the best match with the one with all the others? Which activation pattern is the best match with all of the set of activation patterns? So what you do, remember how we said the way you pick it is you say correlated with this, correlated with that, pick the biggest correlation? That's your classifier. That's all there is to it. So that's what this line here does. It says, we've calculated all the correlations. That's this thing here. Give me the biggest one. Give me the maximum. And maximum actually in Matlab and in general returns 2 values. One is, so I've got 8 numbers here, which are all just different correlations. So one thing it says, what is the highest number in here? So I guess the highest number is 0.6933. And the other is, and which entry gave me the highest number? So in this case, it was the 8th entry gave me the highest number. So this is like the simplest possible classifier. It basically says, find out how well each pattern is correlated with every other pattern, and whichever one's the biggest, pick that one. So this would say, okay, here's a test orientation. It correlates reasonably well with orientation 1, not too well with orientation 2, blah, 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 blah. Oh, it correlates really well with orientation 8. That's the biggest one. So my answer is, I think that this activation pattern was elicited by someone looking at stimulus orientation A. That's it. That's your classification. And so the maximum answer here is best matching index like number 8. So so this is, without actually exaggeration, this is basically what happens in the Haxby paper. So they say, here's the activation for a bunch of different conditions. Take those spatial activation patterns, stretch them out into a line, correlate them with each other, pick the biggest correlation, whichever one the biggest correlation is, say, I think that's what they were looking at. I think that, it's condition number 6 or condition number 8, whatever it was. And that's fMRI decoding. Now, there's lots and lots and lots of kind of tweaks and variations you can do on this. Okay, this is not by any stretch of the imagination the most fancy or powerful classifier, but it's actually a fairly good and robust classifier in many cases. But that's actually how you do it. So this is literally how you would implement in Matlab a very simple classifier, and a fairly effective classifier. And then if you want to actually see how good is it, then you might do it 100 times and add a different bit of noise each time and then plot the result. So, so if you're interested in learning more about the kind of mechanics of how you make these pretty pictures and how you would, for instance, take the average of the results of all of those, then by all means have a look at that. And that gives you these kind of outputs, which are very similar to the kind of pictures that are in the Kamitani Tong paper. So it says that it was really good at decoding this guy. For whatever reason, the noise got in the way, but it was not quite as good at decoding this guy. Yeah, so that's how you do a simple classifier. So this one line here is where the magic happens, and it literally just says, take a bunch of correlations and give me the biggest one. So it's not as complicated as it might seem. So anyway, so thanks very much. I don't want to keep you anymore, but if please ask me any questions that you have either by this or by email, or just now. And if there's anything that's unclear at all that you feel kind of shy about saying in the class, I don't really get that, just post it on Piazza. You can post it anonymously. I know no one's actually done that yet, but please, if you want to do it, the opportunity is there.
Many studies just sort of testing out different. I've seen one or two studies where they sort of test two classifiers against each other. Have there been any just test the whole smorgasbord against a whole bunch of different data sets?
Yeah, there's a few like that. And you can imagine it's a kind of fairly easy way to generate a paper, right? So it doesn't require super much thinking, but you can just kind of, there are some things. I can try and find some references.
But hasn't been why use this?
I don't know. I'm not too worried that someone's going to watch this video on YouTube and say, oh, I got to take that idea. It's something that the reason no one's, I don't really know why, I don't even know, maybe someone has looked at it by now, last time I looked at it. All right, it's kind of messy, it's more complicated, but it's a metric that could potentially involve using spatial information. I think it would be an interesting thing.
So I don't know. You don't want to be in that kind of atmosphere. Yeah, so I'm not sure.
Yeah, I mean, it's not necessarily what's kind of top of my list right now, but it could be something like that. But yeah, there are papers that say, we took the following A classifiers and compared their output and typically similar classifiers actually do very well. But why that is actually sort of a question.
Yeah.
Well, we're going to chat more. Let me just quickly, John.
Oh yeah. So I know you talked about how you the project for taking a little while to get.
Yeah, sorry.
No worries. I was just wanting to know that I may have told you this. I was thinking about using my project as a follow-up to a project I'm currently doing.
Oh, good. I think that's an excellent idea.
It would be fun. I'd love to incorporate fMRI.
I think incorporating your project for this class into what you're kind of doing anyway is a win-win.
Exactly.
Yeah. So I'm super in favor of that.
So would it be possible to either do it sooner or
we should definitely do that. Probably next week is when I plan to try and catch up.
Okay. I'd love to talk to you a little more about it, see if it's actually practical.
Yeah, no, I like that idea a lot. I mean, if you actually want